So today I'm going to present a multimedia presentation on the topic DNA fingerprinting. Now we are all quite familiar with the word fingerprinting. About 100 years ago, people discovered that we could identify people on the basis of the impressions left by the ridges of our fingers. But there was always a quest for a better technique or method to identify and distinguish people. So that method was completed in 1984 by Sir Alex Jeffrey, who discovered DNA fingerprinting. Now, by definition, DNA fingerprinting is a molecular method to identify an individual or to distinguish between any two living organisms of the same species by looking at the unique patterns on their DNA. So, it is a technique that distinguishes people on the basis of the unique patterns present in their DNA. We're quite familiar that DNA is the genetic material of most living organisms. DNA fingerprinting is also called DNA typing or DNA profile. So DNA was used to identify or distinguish between different people to prepare a profile that could be used as an identity for different people. Now, there were two factors which helped in the widespread acceptance of DNA fingerprinting. The first was the uniformity of DNA in an individual, as DNA, we all know, is present in almost all the cells except germ cells. And the other one was the genetic variability, which is found in different organisms. So these were two factors which helped in the acceptance of DNA fingerprinting. Now, brief history, that what were the events that led to the discovery of DNA fingerprinting. As we know, humans have tried different ways of distinguishing people. The first attempt was the ABO blood group system, which was developed in 1900s, early 1900s. People were assigned different blood groups on the basis of the antigens. The other significant discovery was DNA, the discovery of DNA in 1950s. A 3D model of DNA was given. Then there was protein isoform testing, which was developed in 1960s. And finally, in 1980s, DNA fingerprinting was developed. And just a year later, PCR was developed. But the first commercial availability of multiplex STR kits was made possible in 1990s, not before than that. Before than that, it was only in scientific laboratories. Now the question arises, who invented DNA fingerprinting? So DNA fingerprinting was developed by Sir Alex Jeffrey. He was a professor and geneticist at the University of Leicester in the United Kingdom, pioneered the DNA-based identity test. He was a person who pioneered DNA-based testing. In 1984, he developed the first method of DNA fingerprint by hybridization probing using hypovariable dispersed repetitive sequences in human genome. So he was the person who identified that there were repetitive DNA sequences that can be used to distinguish people. Now we'll be talking about the principle on which DNA fingerprinting is based. As we know that on average, 99.9% of the DNA between two individuals is exactly the same. It is just 0.1% that makes us unique. And this 0.1% consists of repetitive DNA that can be used to distinguish people. So DNA fingerprinting uses highly variable repeat sequences and called variable number of tandem repeats. Now, as we've learned in molecular biology, repetitive DNA is also two types. There's tandem repeats and interdispersed DNA. Tandem repeat is also known as satellite DNA. So DNA fingerprinting uses the satellite DNA, which is also various types of different categories that we'll be discussing later on. So, what is DNA, repetitive DNA sequences? They are just nucleotide sequences which are present in multiple copies in our genome, in which the nucleotide sequences of the repeat unit is highly conserved among all individuals except identical twins. But the length and number of these repeats are unique for each individual. variable number of tandem repeats is a type of satellite DNA in which each repeating sequence has 10 to 100 base pairs which are repeated thousands of times. The other type is the short tandem repeats or the microsatellite, where each sequence is of 2 to 6 base pairs which are repeated for about 5 to 200 times. So it is because of this polymorphism that repetitive DNA is used in DNA fingerprinting. And for a matter of fact, the first microsatellite was also discovered by Sir Alex Jeffrey, but was named by Luty and Litt in 1989. Now this was all about the background of DNA fingerprinting. Now we get into the process, what exactly happens in DNA fingerprinting. So before even starting the process, we need to collect biological samples from which DNA is to be extracted. That biological sample can be blood, buccal spear, teeth, hair, semen, saliva, body tissue, sweat, or any other bodily fluids. You can even take the bones of a burned body for DNA fingerprinting, from which DNA can be extracted. Then the first step of DNA fingerprinting is DNA extraction. We've also done DNA extraction in our practical class. DNA extraction is one of the most significant steps of DNA fingerprinting, as good quality and quantity DNA increases the possibilities of getting good results. So obviously, the better the quality of DNA, the better result you get. The purity and quantity of DNA should be 180 and more than 100 nanomoles respectively. So this is the purity and quantity that is required. There are certain techniques that require even really less amount of DNA. And these three are some general methods, extraction methods that can be used in DNA fingerprinting. We've done the third one. We've also used phenol fluoroform DNA extraction method. These are different methods that are used for DNA extraction. Then let's come to the technique and methodology. This is the second step, and it's very important because the technique actually determines what steps you'll be using for DNA fingerprinting. The first technique is restriction, fragment, and polymorphism, which has the acronym RFL. Now, this was the technique that was used by Sir Alex Jeffrey when he developed DNA fingerprinting. So, in this technique, the DNA extracted from the sample can be cut or segmented using specific enzymes called restriction endonuclease that act as molecular scissors by cutting specific regulation sequences and lead to various size DNA fragments. And these are some common restriction enzymes. So what restriction enzymes does is that it recognizes a nucleotide sequence and cut the DNA sample accordingly. So there are different, as you can see in the picture, there are three people and the DNA sequence, the length is different. So it recognizes the repetitive sequences, the number of repetitive sequences are different. In the first there are 3 sequences, 5, 4. So the restriction enzyme will recognize the nucleotide sequence and cut varying lengths of fragments. Now these fragments, restricted DNA fragments, are then separated by using a process called gel electrophoresis. Now gel electrophoresis is a process which separates the DNA fragments depending on the size. The restricted DNA fragments are loaded into the wells and electric current is applied across the gel. This is what we're supposed to do in experiment today. We are supposed to prepare the gel for this. So what happens is that there's a casting tray that has the gel. We are supposed to load the restricted DNA fragments into the wells, as you can see in the picture. There the DNA samples are loaded and there are 2 electrodes on the electrophoretic chamber. These are connected to a power supply when current is passed through it. As you know, the DNA is negative in nature because of the phosphate backbone. So the DNA will move towards the positive end, and the shorter DNAs will move faster than the longer DNA. So as a result, they are all separated, the fragments separated according to their size, which is then visualized. The entire process takes about 30 to 45 minutes. So at the end of the gel electrophoresis, we have already separated the fragments according to their sizes. But gel electrophoresis does not give us a permanent copy. So we need to transfer the fragment that we have got in a memory. And this is done through a process known as sampling. So DNA fragments are transferred from the agarose gel to a nylon nitrocellulose sheet by placing it on the gel and soaking over it. So we use this filter to fragments are double-stranded. So we need to chemically denature those fragments to single-stranded DNA, and then it is transferred on the membrane. The next step is to hybridize a radioactively a fluorescent degradable DNA probe to specific sequences on the membrane. So the next step, what we do is that after transferring the fragments, single-stranded DNA fragments, on a filled-down membrane, we introduce a probe, DNA probe, which is a sequence that has complementary sequences, and it has a radioactive acid or a fluorescent dye labeled DNA probe, which is then this process known as hybridization. It happens in a sealed bag where the membrane is treated with these probes, this radioactive probe or fluorescent dye probe, and it is left for 1 to 16 hours, where the probe attaches itself to the single-stranded DNA, and this is the reason why we are denaturing the DNA before transferring it on a filter. Once the hybridization takes place, the pattern of hybridization is then visualized on X-ray film by autoradiography. So this is the last step. Here you have already got that pattern on the membrane, and you can visualize using autoradiography. This was one technique of doing DNA fingerprinting. The next technique is PCR-based analysis. Now we are already quite familiar with PCR. It is a technique to amplify DNA, small amount of DNA to multiple fold. So what happens that this previous technique that we were using, known as RFLP, it requires a large amount of sample. But PCR was an improvement on that technique, which requires really small amount of sample, which can be amplified into multiple copies. So PCR amplification is a technique that uses an enzyme known as Taq polymerase that has been isolated from a bacterium, which is found in hot spring. DNA primers, DNTP, which stands for deoxynucleoside triphosphates, a template, which is the extracted DNA, and a target region of DNA. So these are the things that we require in PCR. Then these are the 3 steps which are common in all PCR, which is the first denaturing the double-stranded DNA at high temperature is separated, denatured into single-stranded by breaking the hydrogen bond. The second step is annealing. When the temperature is lower, we're taking the DNA primers to attach the template again. Now here the primers, again, the primers attached to the DNA, then extending when the temperature is raised to 72 degrees Celsius, and the new strand of DNA is made by the Taq polymerase. And then what it does is that it adds DNTPs to primers. Here we have labeled DNA inherited diseases, DNA fingerprinting is used to diagnose inherited disorders in both prenatal and newborn babies in hospitals around the world. So, it is very important for diagnosing inherited disorders in both prenatal, when babies still in the womb, and newborn babies. So, what are the disorders that it helps to diagnose? These disorders may include cystic fibrosis, hemophilia, Huntington's disease, familial Alzheimer's, sickle cell anemia, thalassemia, and many others. So, the example that we're taking up would be sickle cell anemia. So, high-class DNA fingerprinting helps in diagnosing disorders. So, what it does is that it uses restriction enzyme, as we've already discussed in RFLP. So, what happens is that there's this restriction enzyme called DDEL. It recognizes, it has this recognition site, but when in sickle cell anemia, there's a point mutation that changes glutamic acid to valine. So it recognizes it cuts it accordingly. In a normal person, that restriction enzyme just cuts it one fragment, but here it cuts two fragments. So this is the basis on which it could be fingerprint. A profile can be prepared on the basis of this, and you could recognize a disease carrier person and a child who carries this disease. The second application is obviously in forensic sciences, in crime scene investigation, criminal verification. So police actually uses this technique to catch the culprit, as we've already seen in CID. This is discussed a lot. So an expert processes the sample and analyzes it against the suspect. As you can see in the picture, a DNA profile is made from all the samples that we can collect from the crime site and is used to create a profile which is then matched to the suspect, and the person who has similar DNA profile is then considered to be the criminal. Different countries have different criteria to use STRs. For example, the US, UK, and India, they have different loci, STR loci that they use. In US it's 13, UK 11, and India is 12 STR markers. These are the markers that are considered in different countries to use for DNA fingerprinting. The third application that we see is in disputed maternity-paternity cases. Now, it is also used to solve these maternity and paternity disputes. A person, as we already know, inherits their VNTRs from their parents. We inherit our VNTRs, the genes from our parents, 50% from our mother, 50% from our father. So this forms the basis of maternity and paternity tests. So the DNA sample of a child is compared with that of alleged father, and similarity is noted when we are performing a paternity test. With this technique, paternity can be confirmed 100%. So a child's DNA profile will be a composite of their parents' DNA profile, and this forms the basis of paternity and paternity test. The same way this test is used for clearing disputed maternity, where a child's DNA profile is compared to that of his mother and father. The fourth application of DNA fingerprinting is genealogy. Another application of DNA fingerprinting is in establishing familial relationships in the area of molecular genealogy. So, DNA fingerprinting also helps us in establishing these familial relationships by using mitochondrial DNA. It helps us to establish these relationships with our ancestors, and this is used a lot in genealogy. But our DNA also contains a record of familial relationships, and DNA analysis can provide clues about what part of the world our ancestors came from. So it also helps us to track our relationships, our ancestors, and this was used when we were searching for Tsar of Russia, when we were searching for his family. This entire process was used, and through this process we were able to track his son and daughter who were burned down in Russia. In one portion of Russia they were burned down, and this technique